Hello fellow students, my name is Ekaterina and I want to invite you to our project of analyzing the genome of bovine herpes virus using bioinformatics. Oh, that's a lot of fancy words here, I know, but actually nothing here is too complicated, and now I'll show you why. Let's start with the definition of a virus. They are the simplest of life forms. In fact, in some classifications they are not even a life form. A virus is a small chemically encoded sequence containing a virus crucial data tightly packed in a protein protective shell, capsid. There are different groups of viruses depending on their method of storing information as a DNA or RNA molecule. Double-strand DNA viruses, like the one in our interest, called herpes virus, can simply use their own cell systems to first read the DNA and transcript it to RNA, and then, according to the genetic code, translate this RNA using ribosomes, creating more viruses in protein shells. While the sequence itself was written down and safely stored in a genome library, no one actually understands what specific parts of it mean. But that's about to change. The goal of a project is to describe this genome. How can we do this? Well, it's a bit like translating an ancient text. When we have the text, we notice that some words or phrases are similar to what we already know, and equivalently, to find the similar to what we know parts in a DNA sequence, we use sequence alignment. There is a lot of math going on, but the principle is very simple. The more similar the mysterious sequence is to the ones we already know, the more we can say about what it may mean. Alternatively, if we already know that the sequences are quite similar, we can try to understand where, when, and why the differences appeared, and maybe make a nice molecular clock out of it. Some words appear a lot of times, but always in a certain context, like the word because. It always means reasoning. Those small but meaningful words equivalents for nucleotide sequences are motifs, like a motif which serves as a start line for ribosomes to start synthesizing protein, the start codon. It's important to identify those DNA keywords and understand what they may mean, and there are some bioinformatics tools for it as well. There are actually a lot of more other interesting things to do, but I think it's already enough excitement for one video. Want to know more? Join me and Aisha at our project and international research school this summer. We'll be waiting for you.
Hi IRS participants, my name is Aisha and you'll be meeting Ekaterina shortly, and we are your IRS tutors. So welcome to IRS 2021. Although this year will be slightly different than past years since we are not together in person, however virtually we'll keep up the same spirit. The project we're proposing to you has to do with, drumroll, bioinformatics and viruses. Yes, viruses. A word you've been hearing non-stop for this past year. But now with this project, you'll be able to uncover the genetic coding of viruses, more specifically the bovine herpes virus, also known as BHV. So what is bioinformatics, you may wonder? Well, it's the ability to use computers and technology to better understand the biological breakdown of genes, proteins, and just about everything biological out there. With bioinformatics, you're able to get down to the genetic code of these viruses and bacteria. I mean, how cool is that? This project will help you understand and visually see the difference between human viruses and those found in animals, while comparing it to the genetic breakdown of the bovine virus. So next time someone asks you the difference, you'll be able to share this cool piece of information with them. So grab your computers and your Wi-Fi routers, along with your spark of your curiosity, and let's get this project started. And Ekaterina will introduce the project to you guys shortly, so stay tuned. Bye!